Hello students, welcome to EPG Pathshala. I am Dietry Ramakrishna from Institute of Liver and Biliary Sciences, Delhi, and today we are going to discuss the module Nutri-Genomics, the latest trends, from the paper Molecular Therapeutics. The major objectives of this particular module includes how genetic variants affect response to the nutrients, how diet influences expression of genes, prenatal and the perinatal nutrition and their effects on the adulthood and the F2 generation, the calorie restriction on the health span. Changes in the dietary and lifestyle habits, especially in developing countries, has posed new challenges for health. The post-genomic era has opened new avenues for better understanding and monitoring the influence of the diet on gene expression, and also based on the genetic makeup of an individual, new dietary regimes can be prescribed by the nutritionist. You are what your mother and grandmother ate. That is, the effects of early diet by mother on the baby has resulted in yet another striking area of research. That is how nutrition can influence the epigenome. Nutrient and certain metabolites can influence the gene methylation and acetylation status, which in turn can influence the gene expression, thereby influencing the health and pathophysiology with respect to disease. One branch of Nutrigenomics focuses on the mechanism by which calorie restriction can be beneficial. Calorie restriction influences the cellular metabolism by changing the NAD to the NADH ratio. This in turn plays an important role in regulating a metabolic regulator called the sirtuins, which help in preventing the oxidative stress and also can influence a number of metabolic processes, thereby influencing the lifespan or extension of longevity of the organism. The talk will briefly overview the emerging focused research areas on nutrition genetic interface, the new area of nutrition epigenome, and the future avenues towards a practical personalized nutrition program. We will study in this module about genetic variants, how food intake of mothers and fathers influences the baby, and the age-old idea that calorie restriction improves the longevity. The major objectives of this particular module includes how genetic variants affect response to the nutrients, how diet influences expression of genes, prenatal and the perinatal nutrition and their effects on the adulthood and the F2 generation, the calorie restriction on the health span. This slide tells about how the genome and the nutrition interact together. The micronutrients and macronutrients from the nutrition helps in a dietary signaling program, which in turn helps in the metabolic reprogramming. Further, the nutrition and how the genetic variants appear determine the prevalence of the diseases such as the cardiovascular disease, type 2 diabetes, cancers, etc. All this was possible because of the unraveling of the Human Genome Project, for which the first draft was available in the year 2001. This slide tells about the three important aspects of the Nutrigenomics, that is the diet, the health, and the genetic makeup. This in fact can be divided into four aspects. Nutrigenetics, that is how genetic variants affect the response to the nutrients. The Nutrigenomics, that is how the diet influences expression of genes. That is how the food and its diet components can influence the expression of the gene to an on or an off state, which in turn determines the health. A new emerging area is that of the Nutri-Epigenomics, that is how the prenatal, perinatal, and also the maternal and paternal diet influences the chromatin of a baby and leading to the changes in the health and disease aspects during its adulthood. Finally, a new era which is emerging is the influence of the gut microbiota on the health and the disease of an organism. So what is Nutrigenetics? To understand Nutrigenetics, we need to understand the complexity of the genome. And this has been much evolved in the post-genomic era. For this, one needs to understand the differences between the mutations and the single nucleotide polymorphisms. And in fact, the single nucleotide polymorphisms, at least in the human genome, exist as 10 to 12 million SNPs. The SNPs have led to the study of what is called as the Genome-Wide Association Study, or the GWAS, where the SNPs are identified in the cases and the controls. The cases means the disease population and the control is the non-disease population. So using next generation sequencing, now the scientists have identified a number of SNPs which are responsible for the disease penetrance. This slide tells about the role of nutrition-related genes and Nutrigenetics in understanding the disease pathology. The influence of human genetic variation on the nutritional requirements in turn is determined by the genetic variant or the single nucleotide polymorphism, and also the consequences of the diet influencing the fixing of a particular gene allele in the population. And these in turn determine the equilibrium between health and disease. This slide provides you the table that tells about the various gene variants that can exist in a human, which in turn can affect the uptake or metabolism of the dietary components. The most important being the folate, the vitamin. This in turn, its metabolism is in turn determined by a number of genes, of which two of them are the most important. For instance, the MTHFR gene and the CBS gene. This schematic shows the process by which the folate or the folic acid is used in the DNA methylation. In fact, there are a number of variants in this pathway for the various genes, of which the most important is the MTHFR 677C to T variant that reduces the enzymatic activity and also divert the availability of the methyl group from the DNA methylation pathway towards the DNA synthesis pathway. This slide tells about the different MTHFR variant and how they affect the folate metabolism. So for instance, if you have a CC genotype, there will not be any loss in the MTHFR activity. However, if you have a CT or a TT type of genotype, this will lead to loss of a function. The folate metabolism is depicted in this picture, and it tells about the differences in the DNA methylation status. Earlier, we had done a small study where the prevalence of the MTHFR polymorphism was done in a South Indian population. And what we found was that this polymorphism results in babies with neural tube defects. The T allele frequency of C677T mutation of MTHFR gene in the South Indian population was established, and this was much lower compared to what was seen in the UK and the US population. Also, we found a gender bias. Females were found to have a higher T allele frequency compared to the males. Further, the homozygous TT females had babies with neural tube defects. So this particular group of individual has to be fed a high folate diet. This slide tells about the polymorphisms which are associated with the lipid metabolism. In fact, if the gene PCSK9, which is pro-protein convertase subtilin or kexin-9 gene, if there is a loss of function of this allele, this is actually protective and the individual will have low amount of cholesterol circulating in the blood. Thereby, the loss of this allele is protective. This proprotein convertase is an enzyme which is encoded on the chromosome 1, and variants of PCSK9 can reduce or increase the circulating cholesterol. The LDL particles are removed from the blood when they bind to the LDLR receptor. As PCSK9 binds to LDLR receptor, it prevents the removal of the LDL particles. Thus, we can understand if there is a loss of this particular allele, it will be protective. Similarly, it has been noted that certain genes such as the APOE, the LDLR, those variants can also have a high risk for the presence of the cholesterol in the circulation. Accordingly, a number of studies are going on where now drugs are being developed to target one or these of these genes so that to prevent the hypercholesterolemia. Yet another important variant noted is for the apolipoprotein E gene. The apolipoprotein is a class of apolipoproteins found in the chylomicrons and the intermediate density lipoproteins, or the IDLs. That is essential for the normal catabolism of triglyceride-rich lipoproteins. The APOE variant in turn influences the health and the metabolism. This slide tells about the APOE gene variants and how they are either protective or deleterious for the lipid metabolism. APOE is a polymorphic gene and has three variants: APOE2, APOE3, APOE4, of which APOE3, with an estimated frequency of 55% in the population, is supposed to be a good allele and is considered for the normal lipid metabolism. However, a variant of APOE4 or an APOE2 results in dysfunctionality of the enzyme, thereby leading to high risks of cardiovascular diseases, Alzheimer's, etc. And this is because of an increased total cholesterol and triglycerides in the circulation. This slide tells about the APOE2 alleles and how that is influenced by the intake of the nutrition. A high-fat diet increases the lipoprotein levels, which in turn increases the cholesterol requirements. Now, research studies have confirmed that the carriers of APOE4 benefit most from the low-fat, high-carb diet, while the E2 carriers benefit most from a high-fat and a low-carbohydrate diet. Diet has also influenced the evolution of genes. Homo sapiens were first hunter-gatherers, and later, as they settled down with farming, there was a huge consumption of not only the vegetables but also animal products like milk, which is rich in lactose. And digestion of the lactose is done by the LCT gene, which codes for the enzyme called the lactase. It has been noted that people with lactose insensitivity have a CC genotype, whereas those with lactose intolerance has a CT or a TT genotype. So if one can do the genotyping, one can actually tell whether the individual is going to be lactose insensitive or a lactose tolerant. So we have covered topic on the Nutrigenetics. Now let's see how the nutrient or the diet can influence the expression of the genes, either through the signaling pathways or through the chromatin remodeling, which is called as the Nutri-Epigenomics. We will first talk about the Nutrigenomics and signaling pathways, and this will be followed by the Nutri-Epigenomics section. This slide shows how diet can modify the signaling pathways. For instance, good nutrition like those which are rich in fibers, which are composed of essential amino acid, and good carbohydrates help in healthy gene expression through various signaling or sensor mechanism, leading to a healthy expression of the genes, which influences a good health. However, a change in diet towards a Western diet with high saturated fats, fructose such as those present in the soft drinks, etc. can lead to metabolic disorders such as the fatty liver disease, the type 2 diabetes, etc. And this is because these kind of nutrients, that is the fructose and high saturated fats, can actually cause a deregulation in the signaling mechanisms, and this leads to the so-called stress signatures associated with metabolic disorders. This slide tells how the micro and the macronutrients influences the cellular factors determining the expression of the genes responsible for their metabolism. For instance, the calcium, which is an important micronutrient, that influences the very well-known calcineurin NF80 or nuclear factor activated transcription factor pathway. And this is important for the T cell activation and the immune response. So now students, you realize looking at this table how the micro and the macronutrient can influences various genes which in turn are linked with the proper body responses. The polyunsaturated fatty acids can activate the cytosolic receptor such as the peroxisome proliferator activated receptor or PAR alpha, which then dimerizes with another receptor called the RXR, and these can then bind to the promoter regions or the response element, leading to activation or expression of certain genes involved in the lipid metabolism. Depending on which tissue the PPAR isoform is expressed, the responses vary. For instance, in liver, it helps in fatty acid metabolism. It helps in the macrophages, the responses with secretion of the cytokines, thereby leading to inflammatory responses. In bones, this pathway activates the osteoclastic activity, and this pathway can also dictate the insulin sensitivity or the resistance. This slide tells about how the different genes and the variants that we had seen earlier in presence of nutrient or the metabolic product can influence the signal transduction pathways, thereby leading to changes in the expression of the genes. Now, this signaling cascade and understanding of these signaling cascade in response to nutrients has given rise to a totally new field which is called as a nutrition systems biology, which helps in categorizing and making of the databases for the different genes which are expressed in response to the nutrients and the metabolites, and also how it influences the gene expression, and this has then resulted in the identification of the molecular biomarkers associated with the disease. Besides the Nutri signaling, another new emerging area is the area of Nutri-Epigenomics. That is, how the nutrition can help in chromatin remodeling. To understand this, we need to understand what an epigenome is. Now, in epigenome, the genome sequence is totally identical. However, on top of this genome, there are certain modifications. And this include the acetylation, the methylation, and the nucleosomal arrangements. And all that on top of the genome is actually called as the epigenome. Now, here is an example how the nutrition will actually help in modifying the epigenome. S-adenosyl methionine is one of the important components in one of the metabolic pathways. SAM, as it is called commonly, is actually a methyl donor. The methylation of the CpG islands can lead to a hypermethylation state, thereby turning off of the gene expression. When this methylation mark is removed, it can lead to the expression of the gene or the on state. With this background, let's now understand how the nutrient and the epigenome interaction takes place. In this work, we had found that women with high folate were less susceptible to infection with human papillomavirus, which is an etiological risk factor of cancer. In another work, we determined that these women with low folate and presence of the high-risk HPV infection show a differential methylation pattern of the gene called the DNMT3L. And the hypomethylation of the DNMT3L makes the expression of the gene, leading to disturbances in the host methylome. So this is one example of how the nutrient such as the folate can interact and influence the disease and the health of an individual. Nutri-Epigenomics has been unraveled using the twin studies, the influence of how maternal or paternal nutrition affects the child when it grows into adult. Not only that, epigenetics effects can be imprinted up to the F2 generation as well, and this is called as the transgenerational effect. Similarly, consuming substrates like alcohol, smoke, nicotine can also influence the genome. The influence of the maternal and the paternal nutrition in turn influences the metabolic disorders in their offspring. A famous study in the Dutch famine study, it was shown that the famine influenced the onset of type 2 diabetes in the coming generations. Similarly, animal experimentation studies have shown that mothers who are undernourished or are not consuming the right diet can influence metabolic syndromes in the babies, which is through epimodulation. I will now discuss a famous animal experimentation study as to how the diet of the mother influences the baby's health and especially the color coat and the obesity. In this famous study, the agouti or the yellow colored mice has no methylation on the agouti locus, and this makes the mice not only a yellow fur but also this mice is obese and it is more prone to cancer. When these agouti pregnant mice were fed a supplement which was rich in zinc, methionine and choline, folate and B12, this kind of a diet is also referred to as a methyl diet. The F1 babies born to the methyl-fed mothers ended up with a thin body structure. Not only that, as you can see, the F1 generation also had a change in their fur color. And this fur color from yellow became black. And this was not only maintained in the F1 generation, but it was also maintained to the F2 generation. So what happens in the methyl diet? The supplements through S-adenosine methionine provides the methyl group, and the enzyme DNMT then methylates the cytosine to 5-methylcytosine at the agouti locus, thereby shutting off the gene expression of the yellow color, resulting not only in black fur but also reducing the body weight to thin, making them less prone to tumors. So you see, what you are is what your mothers ate during the pregnancy. Nutrition epigenomics is just not restricted to maternal food but also is dependent on what the fathers ate. And in this famous study, it was shown that the fathers who were fed with low protein diet, their babies had an altered expression of the genes, especially those involved in the lipid synthesis. And this study shows that you are what your father or your grandfather ate. This is explained in detail in the next slide. Thus, it was noted that fathers fed on low protein diet had offsprings with high risk of metabolic disorders. And this was because the father's nutrition influenced the PPAR gene methylation, turning it off, thereby affecting the lipid metabolism, and thereby the offsprings of the fathers fed on a low protein diet were affected with metabolic diseases and they were much more risk prone. It is also believed that calorie restriction helps in maintaining health, and by calorie restriction one can avoid becoming obese, but more importantly, studies in lower organisms like the worms and the rodents have shown that calorie restriction can also help prolong your lifespan through 3 mechanisms. Number 1, by reducing the insulin signaling, thereby influencing the TOR pathway. The low glucose can also influence the TOR pathway, and more importantly, low calorie can lead to a high NAD to NADH ratio, And NAD in turn activates a deacetylase called sirtuins, which is a very important metabolic regulator protecting the organism from disease and thereby maintaining a healthy lifespan. So now students, you've seen that how the Nutrigenomics influences the longevity, especially by the phenomenon of calorie restriction. This slide gives in a nutshell how calorie restriction can influence the impact of lifespan and how nutrients like resveratrol present in red wine, curcumin, a very important component in Indian diet, can influence the metabolic reprogramming by affecting various signaling events, and this has resulted in the systems biology of nutrition. So students, let us summarize what we have learned in this module. We have learned that the diet can influence our metabolisms. Further, the genes and the variants can influence the dietary output. We learned about the epigenetics of the nutrition. That is, how the maternal and the paternal nutrition can influence the responses in the babies. Further, we learned about the benefits of calorie restriction. Thus, in conclusion, the students, you can now appreciate why the old Indian philosophy emphasized on a balanced diet and cleansing periods of fasting, which is nothing but the calorie restriction. The post-genomic era has taught us that genetic polymorphisms modify the dietary requirements. The genetic profiling can help the clinicians to identify people with metabolic inefficiencies and modulation their dietary intake. The maternal and the infant diet can alter the epigenetic marks in the babies throughout the life, leading to what is called as the transgenerational effects of nutrition. Finally, food choices and lifestyles you can adopt can help in maintaining good health, provide increase in lifespan, prevent disease, thereby helping in healthy aging. Further, your food choices and lifestyle will determine the maternal and the paternal diet can influence the epigenetic marks in the babies. This has led to the development of what is called as the transgenerational effects of the nutrition. So depending on the type of the diet which the mother or the father is consuming will help in putting certain epigenetic marks in the babies. This in turn will determine the metabolic regulation in the babies, and this in turn can either help or prevent the metabolic disorders in the coming generations. Finally, the food choices and the lifestyle you adopt can help in not only maintaining a good health, but as has been seen in the lower organism, that calorie restriction can also increase the lifespan. That is, it can also prevent diseases and thereby helping in healthy aging. So, it is very important to have proper food choices, lifestyle, the duration, and the spacing of the diet to prevent the onset of metabolic disorders. Thank you.